Okay, class. So for today's lab, we will be discussing how pH and temperature affect the functioning of enzymes.
Oh my god, he won't stop talking. I just want to start my lab already.
I know, right?
You know what I could do right now? Some chocolate milk. But it's so hard to make.
Yeah, it would be so much easier to get it straight from the cow instead of having to mix it in all the time.
Oh no, that's something I'm interested in. What if we can genetically modify cows?
Hey, I actually read an article where some scientists are conducting experiments on the total synthesis of designer eukaryotic chromosomes.
Sounds awesome.
Yeah, this sort of research will eventually lead to the feasible engineering of new eukaryotic genomes, including plant and animal genomes, with synthetic chromosomes encoding desired functions.
That's so cool. Tell me more about it.
I can actually show you a really cool YouTube video about it.
Hello, this is Bio Rocks, and I'm your host, Dr. Rob Mitochondria, and this is my co-host, Dr. Matt Scientist.
In this experiment, we use Saccharomyces cerevisiae, a species of yeast that has a genome size of about 12 megabases in 16 chromosomes.
Wait, what's the chromosomes again?
It's a structure that's made up of DNA and proteins, and it carries the cell's hereditary information.
Oh, okay.
The whole yeast genome has about 6,000 genes, but 5,000 are not essential. Our study focuses on the identification and the process of determination of simultaneously dispensable and non-essential genes.
I'm sorry. I wasn't paying attention in class. What's a gene in a genome?
A gene is a hereditary factor that influences a particular trait, and they're expressed through the central dogma. The DNA is used for information storage, and through the process of transcription, it creates mRNA, which then codes for a protein in the process of translation. A genome is all of the hereditary information in an organism.
Yeast chromosome 3 was selected for the synthesis because it is one of the smallest of the 16 yeast chromosomes and controls how yeast cells may and undergo genetic change.
We accomplished this through a process called scrambling. Essentially, we created a synthetic yeast genome in which the changes made to SYN3 resulted in replacements of the stop codons, insertions of LOXP-SYM genes, and deletion of several other genes.
The construction of SYN3 occurred in 3 major steps. First, the building blocks were created using polymerase chain reactions involving 60 to 79 Mer oligonucleotides.
What are oligonucleotides and what are polymerase chain reactions?
Oligonucleotides are a short nucleic acid chain, usually consisting of up to approximately 20 nucleotides. Polymerase chain reactions rapidly synthesizes large quantities of a given DNA segment. DNA polymerase is then used to synthesize 2-stranded DNA from each single strand, and this process is repeated over and over again.
Sounds so interesting. Let's continue.
Second, DNA building blocks were assembled into overlapping mini-chunks. After 11 rounds of transformations, the native sequence of S. cerevisiae 3 was replaced with its SYN3 counterpart.
After creating our new yeast cell, we compared the karyotypes of the SYN3 and the wild type through pulse field gel electrophoresis, which revealed the size reductions between the wild type and the synthetic chromosomes.
The chromosome numbers are indicated to the right, and you can see that the chromosome 3 is not visible in SYN3, and it is reduced in size in SYN3L, or left arm.
What are pulse field gel electrophoresis?
Pulse-field gel electrophoresis is used to separate large fragments of DNA that involves changing the direction of the electron current periodically in order to prevent overlap of the spots from diffusion.
Okay, cool. We can play the video now.
To see if there were any negative effects on the fitness of the strains of the wild type, which is the most common phenotype seen in the wild population, containing SYN3, the colony size, growth curves, and morphology were examined under different temperatures and pH values. These strains were not distinguishable from each other on the colony size test.
In order to determine the loss rate of SYN3, the ability to make MAT alpha cells carrying SYN3 was monitored using the A-like Faker assay. No significant change in the loss frequency was observed between SYN3 and the wild type, but the absolute loss rate was slightly higher for SYN3. Hey, why aren't you guys paying attention? Is that the new Justin Bieber video?
It was a video on bio.
Oh, well, okay, I don't want to see you guys cracking up in class again anymore. I'll get back to work and design an experiment.
Okay.
that time the TA confiscated our phone?
Yeah, I remember that. Thankfully, gene scrambling technology has improved over the past decade, so now that we have the first chocolate cow.
Yay!